Hello, everyone. This video is going to be a review of the topics covered in exam 3.1, which is over chapters 30, 31, and 32. So what I've done is I took the questions and the topics on the exam outline posted on Canvas, and I compiled all the information to answer those questions and what's required of us to know there, and I put into this PowerPoint. So while this is definitely going to be comprehensive and probably a long video, I can't guarantee that there won't be stuff added that our professor talked about in class. So please look at her notes that she posts on Canvas and take a look at the textbook, the PowerPoints on Canvas, things like that. I can't really guarantee that this is every single thing that could be on the exam, but I'm hoping that this review video will give you all the information that you need to know, minus maybe a few specific things that were mentioned once or things like that. So hopefully this will build your understanding of all of the topics, and then we'll just have to see what would be added onto that, but we still have some time. First thing we talked about are shared characteristics of animals. They're all multicellular eukaryotes, lacking a cell wall, and they have extensive extracellular matrix, also known as ECM, which includes proteins specialized for cell-to-cell adhesion and communication. Since they're multicellular, their cells need ways to stick to each other and ways to communicate, and this will especially be important when we talk about animals with tissues. Animals are heterotrophic. They're motile at some point in their life cycle, and other than sponges, they have neurons and muscle cells. So if we're going to talk about multicellularity versus tissues, we'll go through that. Like I said, while cells need a way to adhere to each other and communicate, there is a difference between having many cells and then having specialized cell groups. And then in chapter 30, we talked about embryonic tissue layers, symmetry, the coelom, cephalization, segmentation, and innovations for life on land. So talking about multicellularity versus tissues, like I said, they need a way to stick together. So here's the kinds of problems that both multicellular structures and tissues need to solve. So cell-to-cell adhesion, they have to have a way to stick. They have an extracellular matrix to support the structure of the cells. Cell junctions act as bridges between cells. Cell to extra cellular matrix adhesion. There's proteins involved. We've got cell specialization, so cells for specific functions. But then if we want to talk about tissues, tissues demonstrate a more organized collection of the same kind of cell that forms a functional unit. So if you consider the scale, we can talk about an organism that simply is more than just unicellular. So they're multicellular, they have many cells. And then we can talk about a multicellular organism who has cells specialized for certain functions and processes. And then we can talk about an animal that has tissues, which are more organized collections of cells. With embryonic tissue layers, we talked about diploblasts and triploblasts. Diploblasts have an ectoderm, which comprises and derives the skin and the nervous system. They have an endoderm, which derives the digestive tract and connecting organs. And then triploblasts have an extra layer, which is the mesoderm, which derives the circulatory system, muscle, bone, and other organs. And if you recall, we took some cross sections of how those three layers might be arranged in an organism. So as you can see on the left, we have a diploblast, which simply has an endoderm and an ectoderm. And then the triploblast has an endoderm, an ectoderm, and a mesoderm in the middle. And then these germ layers, these embryonic tissue layers are important because they led to more organized structures. So for example, the ectoderm gives rise to skin cells, neurons, pigment cells. The mesoderm has to do with cardiac muscle, skeletal muscle, with kidneys, blood cells, and muscle, smooth muscle. And then the endoderm, lung cells, thyroid cells, and pancreatic cells. So take a look at some of these specific structures within an organism's body, and then you might have a better understanding and grasp of why it's important to see what these different layers do. And a good feature and an extra outside of class material source to help you understand is this link. Will go to a free online textbook for biology. So that's where I got these pictures from. So go ahead and take a look at these. It has a lot more information there. It might be helpful to clarify these topics. We talked about symmetry. Simply stated, radial symmetry is when an organism has at least two planes of symmetry. Bilateral symmetry is when they have one plane of symmetry. And an extra kind of symmetry that we talked about is biradial symmetry. It just means that an organism has exactly two planes of symmetry. And this will be important to understand cephalization and just as a general way to classify and differentiate different organisms. Next, we talked about the origin of the gut and the coelom. A coelom is a space for oxygen and nutrients to circulate, and it enables organs to move independently. Coelomates have a coelom that's completely lined with mesoderm-derived tissues, so they have a true coelom. Acoelomates do not have a coelom, such as the flatworms that we'll get to later on. And pseudocoeloms, coelomates have a coelom partially lined with mesoderm-derived tissues like roundworms. And again, this comes from that super helpful online textbook that I just mentioned. So on the left, we have a flatworm, which does not have a coelom. So you can see it is a triploblast. It has an ectoderm, a mesoderm, and an endoderm, but other than the digestive cavity on the very middle, it does not have a coelom. In the middle, we have a coelomate, an annelid, which, as you can see, so it's again, it's triploblastic. It has an ectoderm, a mesoderm, and an endoderm. And then its coelom is completely enclosed by mesoderm-derived tissues. And then on the right, we have a nematode, which has a pseudocoelom. Again, triploblastic, but only the body wall is lined with the mesoderm. So when we're talking about the differences between these three, we're really talking about do they have a coelom present, and is it completely surrounded by the mesoderm? One important feature that has diversified organisms and animals is cephalization, which is the evolution of a head and the brain. It's important when we talk about the nervous system. So with bilaterian animals, a greater concentration of nerves at the front of their body means that they can process information better when it comes from the front. And you can see this in a lot of body plans of animals with bilateral symmetry, is that they will have their heads at the front of their bodies, and then they will see from the front, their ears are at the front, their noses, their mouths. So it's really important that they're able to process information and respond to changes and challenges in their environment. And then we also talked about segmentation, which is when a body is broken down into different parts, and this allows for specialization of those different body parts. So when we talk about these features, we're really just talking about how did animals come to respond to different challenges in their environment as we go through evolutionary history. And some of these challenges were found during the water to land transition. So they needed to solve gas exchange issues. How could they prevent water loss, and how could they hold their bodies upright? And to respond to a lot of challenges that comes with living on land, that comes with branching out into different environments and ecological niches, we have these diversification themes. So animals can be incredibly diverse in the ways that they use their sensory organs. When we talk about sensory organs, we talk about your five senses: sight, hearing, taste, smell, touch. But some animals can detect thermal energy, such as pit vipers, for example, with heat sensing pits. And then so the animals can be incredibly diverse in their sensory organs, and that definitely connects to cephalization. Animals can get food sources in a variety of ways. You are probably familiar with the difference between herbivores, carnivores, omnivores. Detrivores consume dead material. There's predators and prey, parasites either within the body, endoparasites, or outside of the body, ectoparasites. They can be suspension or filter feeders, deposit feeders, meaning they take their food from the ground, from the sediment. They can be fluid feeders, meaning they drink their food, they drink up a liquid, or they can be mass feeders. Like I think in the book it had like a picture of a lion eating a piece of, I don't know, a gazelle or a buffalo or something. And then movement, of course. So there's different kinds of skeletons that animals may have, such as a hydrostatic skeleton, which is flexible. An endoskeleton inside the body, such as bones and then spicules and sponges. Arthropods have an exoskeleton on the outsides of their bodies. And then they can have a variety of limbs. And I mean, you know, the sky's the limit when it comes to talking about appendages and limbs. How many limbs do they have? How are they oriented on their body? How is their weight distributed? What about the size? Are they jointed? What's the range of motion look like? Do they have wings? Animals are diverse in the ways of reproduction. There are asexual and sexual reproduction in animals. Parthenogenesis is a form of asexual reproduction where an egg can be unfertilized by sperm and can still develop into an embryo. Viviparous animals give live birth. Oviparous animals lay eggs. Ovoviviparous animals have eggs that develop in their bodies and then they give live birth once those eggs hatch. And life cycles can be diverse as well. So we can have metamorphosis, especially in the arthropods, and we can see indirect versus direct development. Indirect development, we were talking about, I think, incomplete and complete metamorphosis with this as well. So does an animal look completely different from the adult, or does it look like a mini version of the adult? So I encourage you to check that out when we talk about arthropods. The exam outline asks you to sort animals into protostomes, deuterostomes, or neither. I've done this for you here. In the protostomes, so the protostomes, the mouth develops before the anus in embryonic development. We have our rotifers, the platyhelminthes, the annelids, the mollusks, the nematodes, the tardigrades, which are water bears, which you actually don't need to really know about, the onychophores. Some of these words are going to get a lot more difficult for me to pronounce. And the arthropods, so all of these are protostomes. Deuterostomes, we have the echinoderms and the chordates. In embryonic development here, the anus develops before the mouth. And then neither, those were the three groups that we talked about in chapter 30, Porifera, Ctenophora, and Cnidaria. And I'm going to talk about more about these breakdowns as we go through this PowerPoint. So, like I said, in chapter 30, we went over three specific animals of interest, animal groups of interest. Porifera are the sponges. So they're asymmetric, sessile, meaning they don't move, they're attached to their substrate. They can mostly be found in marine environments, although some are freshwater. They're suspension feeders, so they have choanocytes inside, which is a flagellated part of the sponge, which captures food. They create channels of water currents to capture food particles. So that's how they filter feed. Sponges have spicules, which serves as the sponge's skeleton, and spicules are made up of stiff spikes of silica or calcium carbonate. So they can reproduce asexually or sexually. Now we move to the comb jellies. Most are planktonic and predatory, so they don't have stinging cells, as we will see with some of the cnidarians, but they do have sticky cells called coloblasts to trap prey. They can use cilia to move. Most have male and female organs, so you can have self-fertilization there, although fertilization would be external, outside of their bodies. Their larvae are free swimming, and they have a gravity sensing organ. In the cnidarians, we have jellyfish, corals, anemones, and hydroids. Here's where we get the stinging cells called a cnidocyte, which is when the organism ejects a barb that can contain toxins to stun and kill the prey so it can be eaten. Within the cnidarians, we talk about two life forms, a sessile polyp that reproduces asexually, and a free-floating medusa such as this jellyfish here that can reproduce sexually. And anemones and corals only exist as the sessile polyps. Next, we talk about the flatworms, Platyhelminthes. So they do not have a coelom. They are acoelomates. So they lack structures specialized for gas exchange and circulation of oxygen and nutrients. As a result of this, they have a very flat body plan. This picture from the CDC is an adult tapeworm. You can see how very, very long it is and very flat, giving its name the tapeworm. So their flat body plan allows for nutrient and gas diffusion over a large surface area. If you think about it, if they were a lot thicker, it would be much more difficult for nutrients and gas to diffuse and to get to the very middle of themselves. And here we have some organisms within Platyhelminthes. We have Turbellaria or Laria. We've got the tapeworms, which are parasitic, the flukes, and the monogenia or the monogenes, which can be found on fish gills. Next are the annelids, the segmented worms. So they do have a coelom. They have a fully developed digestive tract with a mouth and an anus. And they have a segmented body. So you can see we have an earthworm here. And if you look at these different annelids, you can see there's a lot of diversity in how they look. They may have bristle-like extensions called chetae, which extend from lobe-like appendages called parapodia. And then we have the polychaetes, which have many bristles, oligochaetes, earthworms, and others. And then we have the leeches. Next are the roundworms, also known as the nematodes, which are unsegmented. They have a pseudocoelom, a tube within tube body plan, no appendages. They have an elastic cuticle. Remember, cuticles prevent water loss, and in nematodes, this is molted during growth, and they can be parasitic or they can be free living. Next are the mollusks. So they have a foot, they have a visceral mass, which is where their main organ systems can be located. Mollusks may have a hemocoel, which is an open circulatory system where body fluids bathe organs directly, compared to a closed circulatory system with blood vessels. They may have a radula or radula, which can be found on snails, for example, which is a rasp or a file to scrape food. And then gills, which are a large number of filaments which create a high surface area for gas exchange. Mollusks also have mantles and an outgrowth of a body wall that covers the visceral mass and may excrete a shell. Most terrestrial snails have a mantle which forms an internal lung. And bivalves, the mantle is lined with muscle and forms tubes called siphons. Cephalopods can use jet propulsion to travel very quickly, and the mantle forms a siphon to do that. And next we'll be getting into more about bivalve cephalopods, snails, gastropods. Polyplacophora are the chitons. Did I have a picture of the chiton? This one right here is a chiton, which has an armored shell, as you could have seen. Most are herbivores. They use contractions of their foot to creep along, and they have external fertilization for reproduction. Bivalves are your clams, mussels, scallops, and oysters. Most are suspension feeders or filter feeders. Clams can use their foot to burrow into sediment. Scallops can clap the valves together to move, and then mussels and oysters are sessile, and most reproduce externally outside of their bodies in the water. Gastropods are your snails, slugs, and nudibranchs. I have never heard of this before, but I can almost guarantee you that you don't need to know what this is, because we most commonly see snails and slugs. Again, they have a radula, which is a file, and this allows for many different feeding strategies. They use their foot to creep along. Most reproduce sexually, although some can use parthenogenesis, again, where an unfertilized egg can produce an embryo. Cephalopods it's given their name from their well-developed senses and large brains. If you remember, cephalo refers to the head region, the brain, and this includes your squids, octopuses, nautiluses, and cuttlefish, which are predators. They have the radula, which is a file, and then a beak, which are their jaws functioning like scissors. Again, their mantle can allow them to use jet propulsion to travel away from danger. And sexes are separate, so we have male and female, and fertilization happens internally. Next, you need to know the characteristics and some examples of the phylum Arthropoda, which includes insects, crustaceans, and chelicerates. All arthropods follow a body plan consisting of a segmented body with tagmata. Tagmata are prominent regions that can be segmented. Here we have, for example, a head, a thorax, an abdomen, and the cephalothorax. We'll get to which one has which in a little bit. Arthropods have an exoskeleton made up of chitin and sometimes calcium carbonates in the crustaceans, and they have jointed appendages. In Insecta, we have 3 tagmata: the head, the thorax, and the abdomen. The jointed legs extend from the belly of the thorax, and the wings, if they have them, may extend from the back of the thorax. Typically, we'll see 4 pairs of mouthpart structures on the head, 1 pair of antennae used for touch and smell, 2 or 3 simple eyes, and a pair of compound eyes. Compound eyes you can see in flies, kind of looks like a kaleidoscope. Here we have beetles, butterflies, moths, flies, mosquitoes, gnats, midges, ants, bees, wasps, many bugs, grasshoppers, crickets, dragonflies, damselflies. And over here we have a butterfly. Crustaceans have 2 or 3 tagmata. Some have a structure called a carapace, which protects the head and the thorax, forming a cephalothorax. They have 2 pairs of antennae. Some may have mandibles to bite or chew. And here we see the shrimps, lobsters, crabs, barnacles, isopods, or roly-polies, and the copepods, copepods. These words are too hard. This crab looks like he's seen some stuff, and I don't blame him for looking that way, or her. And with your chelicerates, it's basically your creepy crawlies, spiders, scorpions, ticks, mites, and daddy longlegs. I picked a picture of a scorpion just because I know some people probably have arachnophobia, and frankly, I got tired of looking at spiders, and I hate ticks, so I figured, scorpions, at least there's none that are native to Ohio, as far as I know. I mean, it's pretty cold. So chelicerates have a cephalothorax and an abdomen. They have a structure called chelicerae near the mouth. They've got 4 pairs of legs. Again, 1 pair of chelicerae. They have a pair of pedipalps located just behind the chelicerae, which can be used to manipulate food, transfer sperm, or other functions. And, if you're not squeamish with all of these things, I highly encourage you to look more closely at these structures and compare them between different species, because it's really interesting how they can change. Looking at a lot of the spiders up close, it can be pretty interesting. So, I mean, you'd have to not be arachnophobic. So let me actually go back to see where we're at, just to remind you right here. So we've covered the protostomes. We ended with the arthropods. Now we're moving to the deuterostomes, with the echinoderms and the chordates. Oh, and then at the very beginning, we covered the ones that are neither. Again, this was from chapter 30. So let's go to the echinoderms. In echinoderms, we see radial symmetry in adults. For example, the sea urchin, which I don't know why you would want to hold a sea urchin. If you've ever held a sea urchin, or like somebody who has, this has got to hurt. Like, I mean, stepping on one in the ocean could definitely do some serious damage. I mean, like, it just looks painful. I guess maybe if you're holding it, it would only feel somewhat pokey, but I wouldn't want to hold one. Like, it's cool to look at from afar, but I wouldn't want to hold one. Anyways, echinoderms have an endoskeleton of calcium carbonate. So now we are switching to an endoskeleton, meaning inside. So this is a hard structure located just inside a thin layer of the epidermis, which protects and supports the body. Echinoderms have a water vascular system. So water can flow in and out of the system. And then inside we can have cilia, which causes the fluids to move. And within the echinoderms, we have starfish, sea urchins, and sea cucumbers. Now we can talk about the chordates. We talk about 3 kinds of classifications, but here are all of the features that these 3 classifications have. They have pharyngeal slits or gills, which are pouches in the throat. They have a dorsal hollow nerve cord, which runs the length of their body. The notochord is a rod down the body length, and they have a tail. Cephalochordates are the lancelets, which look like fish, but are invertebrates, so they're not technically fish. These are suspension feeders that pump water into their mouths and trap food in, again, the pharyngeal slits. When we talk about pharyngeal slits slash gills, there can be a lot of differentiation there. Lancelets swim using muscle contractions on either side of their notochord, and they reproduce sexually. The Urochordates are the sea squirts, the salps, and the larvaceans. Suspension feeders again, they pump water into their siphon and across the pharyngeal slits. Adult sea squirts are sessile, but salps and larvaceans can move. Here we talk about their reproduction. So most can release eggs and sperm, but some can retain the eggs internally, or they can reproduce asexually. And these are the closest living relatives to the vertebrates. Now, vertebrates have a spinal cord, which is the dorsal hollow nerve cord, which runs the length of the body. The pharyngeal slits may act as gills, as in fish, or remain a vestigial trait in terrestrial species. Tails may be vestigial. Their notochords can be found in embryos, but these can persist still in cells, such as the intervertebral discs. Very interesting if you look up notochords in humans and essentially the remnants of that in the cells. So now we talk about the difference between, vertebrates are chordates, but vertebrates are super important because when you think about an animal, you're thinking about a vertebrate most of the time. So again, chordates have pharyngeal gill slash slits really. Dorsal hollow nerve cord, notochord, and a tail. But then we can have vertebrates or invertebrates. We talked about invertebrates with the cephalochordates. Vertebrates have vertebra protecting the spinal cord, and they also have a cranium to protect the brain. Vertebrates are animals such as hagfish, lampreys, sharks, rays, bony fishes, amphibians, mammals, reptiles, and birds. The brain has three regions: the forebrain, controlling and interacting with your sense of smell. The midbrain goes with vision. The hindbrain goes with balance and hearing. This is a very simple breakdown for the brain. If you take a psychology class, you should learn a lot more about specific parts of the brain. Or Kent State has neuroscience classes. Very interesting to see the differences in brain structures, forms and functions between animals, and of course in humans specifically. There's a lot of information to be learned there, a lot of things that you can do with that. So very interesting. In the jawed vertebrates, the hindbrain has the cerebellum, which controls fine motor control, and the medulla, which is part of the brainstem. The brainstem controls, for example, your heart rate, your breathing rate, essential functions for life. A super important part of the brain, even though it's very small. It's a very small percentage of the mass of a human brain. And then the forebrain and cerebrum are especially important and prevalent in birds and mammals. We see a lot of personality begin to develop here. With the bony fishes, so we talk about ray-finned fishes, coelacanths, and the lungfishes. Ray-finned fishes are fishes such as goldfish, tuna, and salmon, which have bony rods to support their fins. The importance of learning about fishes is the origin of lung hypothesis, stating that early fishes had an out pocket of the esophagus, so essentially forming kind of an early lung, which was lost later in evolutionary history within the cartilaginous fishes, and then still today used in ray-finned fishes as the swim bladder to help be buoyant and remain upright in the water. And again, the book goes into a lot more detail about why it's important to differentiate all the different vertebrates and understanding the diversification of these animals when we look back at evolutionary history. So I encourage you, if you're interested in that, to read more in the book about it. Next, we talk about the tetrapods, meaning 4 limbs. So these are the reptiles, amphibians, and the mammals. And amphibians, you have your frogs and your toads, salamanders, and your caecilians. Amphibians were the first land tetrapods. Most undergo metamorphosis. A classic example is with frogs. So you have the tadpoles, which come from eggs inside the water. Tadpoles have, they really don't look anything like actual frogs when they're juveniles, but gradually as they mature, they turn into a frog, what you recognize as a frog, really. Across their skin, gases can be exchanged, and most lay eggs in the water. So here we still have animals that are reliant on water for their bodies and also with reproduction. But then when we move into the amniotes, which are all tetrapods except for amphibians, here we have a protective covering on the egg, also known as the amniotic egg, that reduces drying and water loss. The amniotic egg is comprised of the embryo, the yolk sac, and a structure to take the waste of the embryo. So we have mammals, which produce milk with mammary glands. Mammals are endotherms, meaning they can maintain a high body temperature within themselves, and they can have hair or fur. Here we have the egg-laying monotremes, such as the platypus and echidnas. Patch-bearing marsupials like kangaroos, koalas, possums, and wallabies, and placental eutherians. Pretty much every other mammal you can think of. In these eutherians, we have the placenta, which is a structure facilitating the flow of the nutrients from the mother inside the uterus, as well as the flow of waste from the embryo. So it's a more specialized structure to support embryonic development. In mammals and birds, parental care is often extensive. And then the book talks more specifically, since we're talking a little bit about evolution, it focuses a bit on primates and humans, and it breaks that down. Again, it's in the book. It's not too long. I have no clue how much we'll really even need to know about it for the exam, but it's interesting. It's worth knowing, but is it worth knowing just to pass this exam? That's something you have to decide. And finally, just personally, I had to save the best for last because I definitely love reptiles. So reptiles include lizards and snakes, turtles, and tortoises. Your crocodilians, such as crocodiles, alligators, gharials, caimans, also birds. Again, the book talks more about this. Reptiles have scales made up of keratin. Most lay amniotic eggs with the shell, although some reptiles, such as the blue-tongued skink over here, I just had to include a picture of one because I own one. These actually give live birth. And there are reptiles that can reproduce by parthenogenesis, such as the whiptail lizards. I don't know if all of the whiptail lizards reproduce asexually, but a lot of them do, where all of them are female. They have this like mating dance like with another lizard, and then I guess the eggs can turn into embryos. It's kind of weird, but interesting. Reptiles are ectotherms, meaning they cannot regulate their own body temperature, so they have to bask in the sun to generate heat in order to digest their food, and to, even exist. Although a lot of the time they'll still feel cold, and they can go into a period of hibernation called brumation. And then birds have feathers and are endothermic, so they can regulate their body temperature. So that's it. Again, please look at the note outlines for class. Please read the book. And I hope you do well on the exam and have a good day.